There we go.
Okay, I'm interested in aging research because of the general belief that aging is the largest risk factor for many diseases. I'm sure you have already heard this from many speakers from the aging field, that the overarching hypothesis of aging research is that if we can target the aging pathways, we might be able to prevent or treat not just one disease, but many diseases at once. So as you can imagine, this is a very attractive way to battle human diseases. And as you also know, that in the past 2 decades, the aging field made a tremendous progress. Now the hallmarks of aging have been identified. It's a very exciting time for rejuvenation. So the question is aging reversible? And I think this is a very important question, because if aging is not reversible, then there's not much we can do to help improve the health of the elderly individuals. Conversely, if aging is reversible, then which aspects of aging-associated conditions are reversible, and to which extent? So finally, is the cause of aging reversible? And we believe that targeting the cause of aging is more likely to have a long-lasting impact on rejuvenation. Now, it's well accepted in the aging field that the general cause of aging is accumulation of cellular damage. There's therefore much interest in understanding what are the aging causing cellular damage, how do cells respond to this damage, and whether we can target these pathways for rejuvenation. So today I'm going to talk about 3 topics that are related to aging research. And the first one is a stem cell aging and tissue degeneration. Now, you may argue that every cell in our body changes as we age, but stem cells received much attention in aging research because they're responsible for tissue repair and regeneration throughout our entire lifespan. So I'm going to spend quite a bit of time talking about stem cell aging and tissue degeneration. And the second topic is metabolic diseases. And in particular, I'm going to highlight fatty liver disease, which is a very prevalent metabolic disease that currently have no cure. So I'm going to talk about our hypothesis, how this disease progresses. And finally, I'm going to talk about aging-related inflammation. So based on the epidemiological studies, aged individuals have a chronic low-grade inflammation. And this chronic low-grade inflammation has been linked to numerous aging-related conditions and diseases. And in particular, this is a very timely research topic because of the pandemic. As you can imagine, one thing we learned from the pandemic is that COVID-19 is a disease of the immune system because of the aggressive inflammatory response. And we also know that aged individuals are particularly vulnerable to this disease. So it's a thought that aged individuals are particularly vulnerable to this disease because they already have chronic heightened inflammation, even in the absence of infection. So they are particularly sensitive to inflammation-related diseases such as COVID-19. So there's therefore much interest in understanding why these aged individuals have heightened inflammatory response. There's a large body of literature supporting the idea that stem cell function declines during aging, contributing to tissue degeneration and dysfunction in aging tissues. Now, I'll just use the hematopoietic system as an example. during aging, the regenerative capacity of hematopoietic stem cells decreases. And broadly speaking, hematopoietic stem cells can differentiate into the myeloid lineage and the lymphoid lineage. So the myeloid cells include, for example, macrophages, and the lymphoid cells include B cells and T cells. Now, during aging, hematopoietic stem cells are biased towards differentiation into the myeloid cells and less to the lymphoid cells. So this is consistent with the physiological changes of the aging immune system, for example, increased inflammation and compromised immunity. And this is also consistent with the pathological changes of the aging immune system. Now, let me give you one example. Childhood leukemia tend to be of a lymphoid origin, and aged individuals tend to develop leukemia of myeloid origin. So similar observations have been found in other tissues where stem cells are well defined, such as the brain, the muscle, the intestine, and the skin. Now, as another way of introducing this topic, I will remind you of the subject that you have learned in cell biology class, which is the cell cycle checkpoint. And in particular, the restriction point of the cell cycle. Now, as you know, once the cells pass the restriction point, they are committed to proliferation. Now, the restriction point is particularly relevant to stem cells, which frequently exit cell cycle and enter quiescence. You may think that the stem cells who are capable of proliferating, so they may be proliferating all the time, but it's actually not the case. Once animals enter the adulthood, the majority of the stem cells are actually quiescent. Now, using hematopoietic stem cells as example, under homeostatic condition, actually 90% of them are actually quiescent. And it's a thought that maintaining quiescent is important to preserve the integrity of the stem cells and preserving the stem cell pool. Now, for quiescent stem cells, they are relatively metabolically inactive, and they also have a less mitochondria. They primarily rely on glycolysis for energy production. Now, this is important because it bypasses the use of mitochondrial oxyphos and bypasses the production of metabolic waste, such as reactive oxygen species. Now, stem cells are particularly sensitive to oxidative stress, reactive oxygen species. So therefore it's beneficial for stem cells to avoid mitochondria, rely on glycolysis when they don't have to. This is possible for quiescent stem cells, which have lower energy demand. As you know, mitochondria oxphos generate a lot more ATP compared to glycolysis. But for quiescent stem cells, they are certainly sufficient to rely on glycolysis for energy production. Now, when stem cells exit quiescence and enter proliferation, there's an increase in mitochondrial biogenesis. And this is coupled to the switch from glycolysis to mitochondrial oxyphos to meet the increasing energy demand of proliferating stem cells. Now, because one major event during this transition from quiescence to proliferation is mitochondrial biogenesis, it makes sense that a major cellular event that needs to be monitored during this cell cycle checkpoint, the restriction point, is the mitochondrial health. And the failure to do so can lead to the stem cell death and the depletion of the stem cell pool. So when I started my lab a number of years ago, we were really intrigued by a study published by Peggy Goodell's lab, who compared the transcriptional profiling of young and old hematopoietic stem cells. And among the list of genes that are differentially expressed in hematopoietic stem cells during aging, are three sirtuins, 32, 33, and 37. Now, for those of you who are not familiar with the Sirtuin family, there are 7 of them in mammals. They have a deacetylase activity. And for these 3 Sirtuins, they are localized in different cellular locations. And at that time, the field was very much focusing on SIRT1 for a number of reasons, and all the other Sirtuins were pretty much overshadowed. So we thought it was a good opportunity to learn something new about Sirtuin biology, and certainly a good opportunity to learn something about stem cell aging. So we essentially asked the naive question, what do these 3 Sirtuins do in hematopoietic stem cells during aging? So in general, we combine the approach of biochemistry and mouse genetics. So using biochemistry, we were able to map the biological pathways regulated by these Sirtuins, and then we move into the mouse models and ask how these pathways operate in the hematopoietic stem cell compartment.
Danica, quick question regarding that. Do we know of a common regulatory factor or a set of factors that are responsible for downregulation of these genes as we age?
Yeah, that's a very good question. That's actually something we are interested in identifying at this moment. We tried a few suspects, but turned out we were wrong. So we're continuing looking. Hopefully, I can update you at some point in the near future.
Thank you.
Yeah. So SIRT3 is in the mitochondria, and it has deacetylase activity. Now, turns out lots of proteins in the cells are modified by acetylation, and in particular inside the mitochondria, about 15 to 30% of them are modified by acetylation, and the SIRT3 is the major deacetylase that regulates acetylation of mitochondrial proteins. And one substrate we identified for SIRT3 is a superoxide dismutase 2, which is also modified by acetylation in cells. And when it's modified by acetylation, it has reduced enzymatic activity. So SALT2 stands for superoxide dismutase 2. It's a major antioxidant inside the mitochondria. Now, when it's deacetylated by SIRT3, its enzymatic activity is increased. And therefore, a major cellular function of SIRT3 is to reduce oxidative stress. SIRT7 is inside the nucleus. It has a histone deacetylase activity. So what we found is that SIRT7 is recruited to the promoters of mitochondrial ribosomal proteins and mitochondrial transcription factors through its interaction with the transcription factor NRF1. Now, because it's a histone deacetylase, as you know, it suppresses gene expression of mitochondrial ribosomal proteins and mitochondrial translation factors, and therefore suppresses mitochondrial translation. Now, this is particularly important when cells are experiencing mitochondrial protein folding stress. Now, I'm sure you are very familiar with the ER stress, but mitochondrial protein folding stress is a similar concept, except that it is a protein folding stress inside the mitochondria. The general response to mitochondrial protein folding stress is very similar to ER stress. So on one arm, it will trigger the suppression of translation, which makes sense because when cells are experiencing protein folding stress, you want to reduce translation and reduce the production of new proteins when cells are already experiencing the protein folding stress. So this will give cells more time to recover from a protein folding stress. And at the same time, it also induces the production of chaperones and heat shock proteins that help with the protein folding.
Is this mitochondrial protein stress related to the proteins that are expressed from mitochondrial genome, or are these nuclear proteins that are transported by?
These are all nuclear proteins. So there's, for example, the Hitchcock, HSP60, HSP10, and the CLPP. So all of these are encoded by the nuclear genome. And 37, and of course, this is all happening inside the nucleus.
So the question is this the consequences of ER stress, or does it contribute to it? I mean, these are nuclear proteins which are misfolded and they're causing mitochondrial dysfunction.
So they are induced by the stress, and then they help recover help with the protein folding to reduce the stress.
Right, but I was asking if mitochondrial stress, mitochondrial protein misfolding stress, is it something that happens along with the ER stress? Is there like a overlap between?
Oh, I see. Whether there's an overlap, I'm not aware of studies directly looking at the crosstalk between mitochondrial stress and ER stress mitochondrial protein folding stress and the ER stress at the same time. But I would imagine, in principle, it can be possible, right? If for example, under certain conditions, I would imagine it's possible, but I haven't really seen data or studies specifically looking at the crosstalk in terms of the protein folding stress. But we all know that there's a lot of crosstalk between ER and the mitochondria for sure.
Right, and maybe progression of senescence or senescence induction may be a good model to study something like this.
Yeah, for sure.
Cause senescence. So.
Yep, that's a good idea. And SIRT2 is inside the cytosol. It also has a deacetylase activity. And one substrate we identified is NLRP3. Now, as you know, NLRP3 forms the NLRP3 inflammasome that produces inflammatory cytokines such as IL-1 beta and IL-18. Now, acetylation of NLRP3 facilitates the formation of NLRP3 inflammasome and promote the production of inflammatory cytokines. Now, by deacetylating NLRP3, SIRT2 is able to suppress the activity of NLRP3 inflammasome and reduce the production of inflammatory cytokines. And we have also shown that in animal models, reduces aging-related inflammation. Now, with these pathways that we identified, we moved into the mouse models with these 3 Sirtuins deleted individually, and we characterized the hematopoietic stem cell aging-related phenotypes. As I mentioned earlier, key hematopoietic stem cell aging phenotypes include reduced regenerative capacity per cell and myeloid biased differentiation. So when we characterized the SIRT2 knockout mice, we found that they have age-dependent hematopoietic stem cell defects, meaning that when these mice were young, they were essentially normal. So we couldn't really see much defects. However, at a 2-year-old, which is really old for mice, we began to see more exacerbated hematopoietic stem cell aging phenotype. So compared to the age-matched wild-type mice, they have a further reduced regenerative capacity, and they have a further exacerbated myeloid bias differentiation. Okay, so so for students in the future, when you are working on aging-related projects, let's say if you have a hypothesis and you wanted to test it in the animal models, but somehow the mice did not really show the phenotype you are looking for. So I would recommend you age the mice and then see whether you can actually see the phenotype you are hoping for. So this is really a lesson we learned in our early days when we were still not as good in terms of learning from mouse models. So we were able to actually really see the phenotype after the mice become older. So I think it's actually further confirming these regulators are indeed aging related regulators, right? Because you can only see the phenotype at an older age, but not a young age. And similarly, we saw the same, very similar phenotype for 33 knockout mice. And again, we did not see any defects in hematopoietic stem cells when the knockout mice were at a young age, but we were able to see further exacerbated aging phenotypes at a 2-year-old. Now for 37, it's a slightly different. So we saw premature hematopoietic stem cell aging phenotype, meaning that at a young age, they already have this aging phenotype, including reduced regenerative capacity and a myeloid biased differentiation. Now, this is a question I'm most excited about. Can these pathways be targeted to reverse stem cell aging? Now, to answer this question, we did the following experiments. So we isolated hematopoietic stem cells from a 2-year-old mice, and we overexpressed either 32, 33, or 37 to this aged hematopoietic stem cells through lentiviral transduction. And we then transplanted back into recipient mice, so bring them back into animals through bone marrow transplantation. And this will allow us to evaluate hematopoietic stem cell function in vivo. So essentially, overexpressing either 32, 33, or 37, we were able to improve the function of aged hematopoietic stem cells. And we did similar experiments to inactivate the NLRP3 inflammasome, either by knocking down NLRP3 or knocking down caspase-1, which forms a complex with NLRP3. And again, using 2-year-old hematopoietic stem cells, and we were able to show that either knocking down NLRP3 or caspase-1 were able to reverse the hematopoietic stem cell aging phenotype. So this is our working model. As the stem cells exit quiescence and enter proliferation and enter the cell cycle, there's an increase in mitochondrial biogenesis. And this is associated with increased mitochondrial stresses. And what 33 and 37 are doing is to suppress the mitochondrial stresses and help cells to return to quiescence and recover from the stress. And failure to do so can lead to cell death mediated by the NLRP3 inflammasome. And the 32 can prevent the death of hematopoietic stem cells by suppressing the NLRP3 inflammasome. Now, during aging, because this 3 Sirtuin expression was reduced, essentially the aged stem cells lose the protection of these Sirtuins. This results in increased mitochondrial stresses that we see in aged hematopoietic stem cells and increased sensitivity to cell death. And we were able to show that these pathways may be targeted to reverse hematopoietic stem cell aging. Now, so far, all these work I have shown you was done in hematopoietic stem cells. So the question is, are these mechanisms conserved across tissues? As I mentioned earlier, there are several tissues that have a very well-defined stem cells. Now, to answer this question, we recently ventured into neural stem cells. Now, as you know, there are 2 brain areas that have a very well-defined neural stem cell pool. One is in the dentate gyrus, which is responsible for learning and memory. And that is the neural stem cells we're focusing on. So we did a single cell RNA sequencing analysis comparing young and old dentate gyrus. And what we found was that in activated neural stem cells, neuron progenitor cells population, the expression of mitochondria-related genes was upregulated, and cell cycle-related genes was upregulated, and the ribosomal proteins was downregulated. Now, this is interesting. So the increased mitochondria genes and cell cycle genes suggest that aged activated neural stem cells may be accompanied by dysregulation of mitochondrial activity and proliferation. And the reduced ribosomal proteins may suggest that these cells are experiencing protein folding stress. And given that a mitochondria is particularly activated, it may suggest that mitochondria protein folding stress is also activated. Now, to test that, we specifically look at the expression of HSP60, which is a H-shell protein inside the mitochondria and is a marker for mitochondrial protein folding stress. Now, based on the single-cell RNA sequencing data, we found that in quiescent neural stem cell and astrocyte population, the expression of HSP60 is relatively low. And in activated neural stem cell, neural progenitor cell population, HSP60 expression is much increased. So this is consistent with the idea that during stem cell activation, there's an increase in mitochondria biogenesis and increase in mitochondrial stress. Now, during aging, just comparing young and old dentagerous, the expression of HSP60 is not changed in quiescent neural stem cell astrocyte cell population, but it's further increased in activated neural stem cell, neural progenitor cell population, suggesting that mitochondrial protein folding stress is increased during aging in activated neural stem cell population. Now, this is at the mRNA level. Now, what about at the protein level? So we did an immunostaining to test that. So we labeled HSP60, and we also labeled activated neural stem cells with MCM2 and quiescent neural stem cell astrocyte population, we used GFAP. So we were able to show that in this quiescent astrocyte population, the expression of HSP60 is relatively low compared to the activated neural stem cell population. During aging, there's no change in HSP60 in the quiescent neural stem cell astrocyte population. However, it's further increased in activated neural stem cell population. So this is consistent with what we found at the mRNA level based on the single-cell RNA sequencing data. Now, because the dentagerous is really packed, and we were just afraid that at this level of a staining, it does not give us the resolution to really look at the colocalization of HSP60 and MCM2. So we did a close-up imaging and a 3D reconstitution of the staining.
I'm sorry.
Yes.
Just to clarify my own thinking about what's going on in these experiments. So you don't imagine that the same activated NSCs in the old animal are, the same population of NSCs in the young animal. I would assume that the NSCs that were activated in youth are long since either, integrated into a neuronal circuit or have passed on, whereas the ones that are activated in the old animals were quiescent, a week previously and are just newly activated at the moment of the experiment. So is the assumption here that you see in the old animal similarly quiescent NSCs until the moment they are activated, and then suddenly under activated conditions they undergo this stress that the young animal does not? Is that the way to understand what's happening here?
I think that's a very good question. I don't think there's a clear-cut answer to that, right? So stem cells, by definition, they can self-renew and differentiate, right? So even for the stem cells in the old animals, it does not necessarily mean that they were just recently activated from quiescent stem cells. Some of them could have lasted a longer time, right? Because they are able to self-renew and differentiate, right? Because typically the typical model is that stem cells has this asymmetric cell division, and one gives rise to differentiated cells and one maintains stem cells.
But between cycles, they're quiescent, and then they undergo a cycle of activation where one cell is a self-renewing daughter and the other cell is the differentiating daughter, and then it goes back into quiescence. So it's not as if there is I mean, correct me if I'm wrong, but it's not as if there is a subset of cells that are constantly activated. Rather, it is that the stem cell is quiescent and then activated and quiescent and then activated and so forth, unless it's a daughter lineage that goes on to become a differentiated NSC, right? So,
right.
So while they're quiescent, I'm reading this to mean they remain free of stress, but when they activate, they suddenly undergo this stress, whereas under the same conditions, when it activates as a young cell, it does not. So is that the right way to think about it?
I see your point. So as I mentioned earlier, I'm fairly new to neural stem cells, and this is our first neural stem cell study. So I think I would need to talk to some experts on this to really think about this. But based on the literature, it is definitely very well established. If you just compare this neural stem cells from young and old animals, there are substantial differences in terms of their function, for example, and in terms of their number. So the neural stem cell number is reduced, and the neurogenesis is reduced. So indicating reduced
the
neural stem cell
intuition
function
would be that there is some existing damage inside the NSC, the aged NSC, even during its quiescent phase, and at least as regards the stress marker, which I guess is a functional marker rather than the structural marker, you're not seeing that in the aged quiescent cells, right?
I guess another way to look at this is, well, first of all, even if they can go, cycle back and forth between quiescence and proliferation, it doesn't really mean that every time they go back to quiescence, all the stress is fixed throughout the entire lifespan, right? And another way of looking
as far as this marker goes, though, you are not seeing that they are under stress as regards at least this assay when they're in the quiescent state.
Yes. So because they have a reduced number of mitochondria, and once their mitochondria biogenesis is activated, and presumably it's also plausible that the mitochondrial stress is not really fixed. And once the mitochondria biogenesis is activated, they will still have a stressed mitochondria. That's one possibility. And another possibility is this the neurogenic niche, right? So lots of signal that actually comes from, just 1 second, my computer is out of battery, so let me connect my computer very quickly. I very apologize. Okay. So another possibility is from the niche. So it's the aging neurogenic niche that also causes the stress in the neural stem cells of old animals.
Once they're activated.
Yeah. Yeah, that's a good question. So we also did a close-up imaging and a 3D reconstitution, which allow us to have a high resolution imaging of the co-localization between HSP 60 staining and MCM2 staining. So we only looked at the HSP60 signal that is in close proximity of MCM2 signal. So we were able to show that the HSP60 signal is indeed much higher in activated neural stem cells from old mice compared to young mice. Now, after showing that mitochondrial protein folding stress increases during aging in activated neural stem cells, and we asked the question, so how does this mitochondrial protein folding stress affect the function or maintenance of neural stem cells? Now, to address that, we characterized 37 knockout mice for neural stem cells and neurogenesis in the dentate gyrus, because 37 is a regulator of mitochondrial protein folding stress. And we first did immunostaining of HSP60 and showed that indeed HSP60 expression is increased in activated neural stem cells of 37 knockout mice, and however, it's not increased in quiescent neural stem cell and astrocyte population. So this confirms that 37 indeed suppresses mitochondrial protein folding stress. And in 37 knockout dentate gyrus, the number of SOX2 positive cells is reduced, and SOX2 is frequently used as a marker for neuroprogenitor cells, although it labels other cells as well, but it's still one of the frequently used markers for neural stem progenitor cell population. And we also did a BRDU labeling experiment, long-term BRDU labeling for 4 weeks. And we were able to show that in 37 knockout mice, the number of BRDU positive cells in the dentate gyrus was reduced in the knockout mice, and suggesting that 37 promotes the maintenance of neural stem cells and neurogenesis in the dentate gyrus. And so then we decided to look at neural activity in 37 knockout mice in the dentate gyrus and the CA1 region. So to do this, we collaborated with our colleague at UC Berkeley, Naji, who built the 2-photon microscope. Now, even with laser, it cannot really penetrate into the dentate gyrus. So what Chilling, a postdoc in my lab, did was that she spent nearly a year to perfect the surgery. She gradually and very carefully removed the layers of the brain tissue that is above the dentate gyrus. So this allows laser to penetrate into the dentate gyrus and allow us to image this brain region. And she injected a virus expressing GCAMP, which is a calcium probe that allow us to label neural activity. So what Chilling found was that the neural activity in both the CA1 region and the dentate gyrus was increased in 37 knockout mice. So this activity is actually not beneficial. So it's a neural hyperactivity, which has been observed in old animals and old humans, as well as humans with dementia and Alzheimer's disease. So we are very excited about this finding, that 37 is actually able to suppress neural hyperactivity. And of course, what we are most excited about is whether 37 actually promotes learning and memory, because both neurogenesis and reduced neurogenesis, neural hyperactivity have been shown to contribute to loss of cognition. So we did a Morris water maze test for 37 knockout mice, and we were able to show that 37 knockout mice spend a longer time to find the platform. And for wild-type animals, the they prefer to stay in the target quadrant or the quadrant where the platform is. However, for 37 knockout mice, they lost the preference. So it suggests that 37 promotes cognition. Now, finally, we asked whether we can use 37 or suppressing mitochondrial protein folding stress to improve aging-related neurogenesis and cognition. So what we did was following. So we did a stereotaxic injection of lentivirus expressing 37 in 10-month-old mice. And 1 month later, we did a BRDU labeling. And another month later, we did a neurogenesis analysis. So in this case, we use a 10-month-old mice because as we use a much older mice, it's become much, much harder to reverse the cognition. So in this case, when we overexpress 37, we did not see change in the number of SOX2 positive cells in the dentate gyrus, which may suggest that once this neural stem cell or neural progenitor cells were lost during aging, maybe that loss of a stem cell number is not reversible. However, when we did a 4-week BRDU labeling, we were able to see increased BRDU positive cells in the dentate gyrus of mice overexpressing 37. So the function of neural stem cells in older mice may be reversible. And we also did water maze test, and 37 overexpression was able to improve cognition, even though the increase is pretty mild.
Would you expect results to be much more pronounced if you were to knock in SIRT7 in the knockout mice instead? Because you're overexpressing this over and above physiological levels, right?
Yes. So we have not done that experiment. Yeah, for sure, the knock-in experiment would be much more robust. And so yes, we have not done that experiment. Now, what's the therapeutic potential of this pathway? So I guess another way of answering your question is right here. So all these Sirtuins are NAD-dependent enzymes. So, so potentially one way of activating all 3 Sirtuins at the same time is boosting NAD level. And in fact, there's evidence that the NAD level reduces during aging, so which may provide additional mechanism to inactivate Sirtuins during aging, which is, as I mentioned earlier, the expression of several Sirtuins decreases during aging, but the NAD level also decreases with aging. So these are just additional mechanism to inactivate Sirtuins during aging. So one possibility is that if we boost NAD, we might be able to activate all these pathways at the same time and potentially protect mitochondrial health and stem cell health. So in fact, there are already some evidence supporting this idea. This is just one example. NAD replenishment using nicotinamide riboside improves mitochondrial health, stem cell function, and extends lifespan in mice. And this is another example using NAD booster, nicotinamide riboside. And in this case, they specifically tested hematopoietic stem cells from both mice and human, and they were able to show improvement. Okay, any questions before I move on to the next topic? Okay. So fatty liver disease, as I mentioned, is very prevalent. It affects about 1/3 of adults in developed countries. And currently there's no cure. So many companies are working to identify a drug for this disease. Now, in the literature, there are a number of studies suggesting that this fatty liver disease or NAFLD patients are associated with reduced IGF-1 level. Now, for this audience, you don't need my introduction about IGF-1, and IGF-1 has been studied in the aging field for a long time, and there's a rich knowledge about IGF-1 and this pathway. So we thought it might be a good idea just to really build on our knowledge in aging biology and use that to help us understand the fatty liver disease. Now, here's a question. So in this scenario I just explained, the low IGF-1 level is associated with NAFL patients. And in particular, it's associated with the severity of fibrosis. However, we also know that calorie restriction so far is the only intervention for fatty liver disease. And we also know that calorie restriction reduces IGF-1 signaling. So in the second scenario, reduced IGF-1 signaling is actually beneficial for this disease. So how can we reconcile this paradox? So to address this, we are asking the following questions. First, we were trying to understand how does IGF-1 signaling become dysregulated in NAFLD? And secondly, how does dysregulated IGF-1 signaling control liver damage in NAFLD patients?
I would say that leaving the IGF-1 question aside, it is really important to conceptually distinguish what you're doing with NAFLD patients, which is just correcting the excessive energy that is driving pathology with calorie restriction, real anti-aging calorie restriction, which is a restriction of calorie intake beneath normal physiological needs.
So what's the last thing you said? What's the physiological what?
I'm not sure where you left off there, so I'll just repeat myself. That it's important to conceptually distinguish between what you're doing with NAFLD patients, which is just, restricting the excess calories over and above physiological needs that are driving steatosis in the liver, versus actual anti-aging calorie restriction, which is restricting energy beneath normal physiological needs.
Yes, I agree with you. I think hopefully I can answer your question at the end of this session. So we think we have some idea, and I think it's related to the anti-aging aspect. So to answer this question, we took advantage of a mouse model we have developed, which is a 37 knockout mice that spontaneously develop fatty liver, and it has a progress to NASH because it comes with inflammation and apoptosis and liver damage. And we have also figured out previously that 37 prevents the development of fatty liver by suppressing ER stress, which is a driver from steatosis to NASH. Now, we did a single-cell RNA sequencing analysis comparing wild-type and 37 knockout liver, and we found that IGF-1 expression is suppressed in hepatocytes of 37 knockout mice. And the plasma IGF-1 level is also reduced in 37 knockout mice. So essentially, this knockout model has a reduced IGF-1 level and also develop fatty liver. So, so we can actually use this mouse model to address the questions that I raised earlier. Now, just keep a long story short, because we know 37 suppresses ER stress, so we ask whether ER stress drives the suppression of the somatotropic axis or the IGF-1 signaling pathway. And so to answer that, we knock down MYC because we know 37 suppresses MYC to reduce ER stress. So we knock down MYC in the livers of wild-type or 37 knockout mice, which rescued ER stress in the livers of 37 knockout mice, and also rescued the expression of IGF-1 in the liver and the plasma IGF-1 level. So this suggests that ER stress triggers the suppression of the somatotropic axis. And we were also able to ask, so how does ER stress suppress the somatotropic axis. So we know that ER stress triggers transcriptional response of number of genes that are related to ER stress response. So we did the analysis of a chip sequencing data for a number of transcription factors related to ER stress response. And we found that ATF3 binds to the promoters or enhancers of a number of IGF-related genes. And when we knock down ATF3, we were able to show that for control cells, the expression of somatotropic genes was reduced in response to ER stress and inducer, but in ATF3 knockdown cells, this response was blunted. So suggesting that ER stress induces ATF3 to suppress the somatotropic genes. Now, we wanted to ask the question, how does ER stress suppress the somatotropic axis to control liver damage in novel development? So we knocked down ATF3 in wild type and 37 knockout liver. And so this, and we showed in data that I'm not including here, we showed that it rescued the somatotropic axis. And what we found was that the inflammation in the liver was increased, but the fibrosis was suppressed. Now, this is interesting. So in almost all the studies I have seen so far, essentially, when they have an intervention, they're almost all protective or giving disadvantages effects. But I think this is the first study I know of, a sort of given mixed benefits. So some are better and some phenotypes get worse in terms of fatty liver, right? So in terms of the treatment of fatty liver, essentially we're trying to target the two aspects. One is liver inflammation, another is fibrosis. So in this case, it seems like we can't really get all the benefits. So we can only rescue fibrosis, but not inflammation. So because this disease has been studied for quite a while and we're trying to find a cure and haven't been successful, there have been some talking about combination therapies. And I think our study actually provides some support of this idea, at least, for example, perhaps combining IGF-1 with other interventions to accomplish this goal. So hopefully that's the case. And so this is essentially the working hypothesis we have. During the progression from steatosis to NASH, ER stress suppresses the somatotropic axis. Now, from what we learned in aging studied, suppression of the somatotropic axis reduces cell death and also reduces cell proliferation. So it's actually a stress response to prevent cell death. And of course, by suppressing cell proliferation, it is also a stress response, right? So when cells are experiencing stress, they tend to slow down growth and proliferation and give them time to recover from the stress. Now, because this is a reduced cell death, and this will also lead to reduced inflammation. Now, this reduced hepatocyte proliferation is not beneficial in terms of liver physiology because it will reduce the parenchymal repair that is mediated by hepatocytes. Now, when there's no sufficient parenchymal repair, the liver has to depend on the repair by stellate cells, which is manifested as a fibrosis. So this, I think, can explain why in novel patients, there's association between suppressed somatotropic axis and the severity of a fibrosis. And I think this can also explain why calorie restriction can actually suppress the somatotropic axis and be beneficial for this disease, at least at the very early stage, because at the very early stage, by suppressing the somatotroph axis, it prevents cell death and prevents further liver damage. So therefore, at least at the early stage, it is beneficial.
A quick question regarding that. When you say it suppresses inflammation, what do you actually mean by that? Is it the suppression of TNF alpha, TGF beta pathways?
Yes. So in general, we just by looking at the expression of inflammatory cytokines in the liver. So because inflammation is triggered by cell death, so we think that by reducing cell death, we could also reduce the occurrence of the inflammation.
Right. So the reason I was asking this is because we know that the stellate cell function in the liver is absolutely essential for resolution of fibrosis, and TNF alpha signaling actually is important for those cells to function, for the phagocytic function. So is that like an arm also that is in play that we may want to consider, like we are by suppressing inflammation, you're also suppressing the natural phagocytic function of the macrophage-like cells in the liver. And that, again, not resolution fibrosis or sustained fibrosis.
Yeah, we have not really gone that far yet. So this is as far as we got. So this is a work in progress, and we are definitely looking at, we're interested in further collaboration to look at this even further.
So thank you.
So I think it's at 2 o'clock right now. So do you want me to continue, or this is a?
Yes, that makes sense to get 2 reviews. First, take for another 10-15 minutes. Okay. I think we got another 10-15 minutes before the students need to depart for their other experiments. So you can keep going.
Okay, sounds good. I will try to be quick. So the next topic is aging-related inflammation. So we found that when substrate of SIRT2 is NLRP3 inflammasome, and we found these lysine residues that are targeted by SIRT2 for deacetylation are critical for the NLRP3 interaction and NLRP3 ASC interaction. So I'm going to more focusing on aging-related data. So when we characterize the SIRT2 knockout mice, at a young age, they have a normal amount of inflammatory cytokine IL-18 in the plasma, and we also did look at their glucose homeostasis by GTT assay. They are pretty much normal. However, at an old age, so they are 2 years old, and we saw that this SIRT2 knockout mice has further increased IL-18 in the plasma, and they are more glucose intolerant. So then we wanted to find a way to ask, okay, so we know SIRT2 prevents the aging-related inflammation. So can we actually target SIRT2 to reverse aging-related inflammation? So how can we do that? So then a postdoc decided that he wanted to set up this co-culture system with macrophages and a piece of metabolic tissue. So this is fine. However, there's one challenge, which is for macrophages, especially, so the primary macrophages, they are very difficult to manipulate genetically, right? So then we decided, okay, so can we actually find a way to immortalize them? But then the question is, after immortalization, whether they still have this memory of old age. So essentially, we immortalize the macrophages from young and old mice. So in this case, we actually immortalize the myeloid progenitors with HOXB8 and then differentiate them into macrophages. So I will just simplify it and call them immortalized macrophages. And then we test whether they still have this memory of old age after immortalization. So this is the experiment using primary macrophages and comparing this young and old primary macrophages, and we saw increased NLRP3 inflammasome activation. And in the immortalized macrophages, and we still see increased NLRP3 inflammasome activation. So even after this immortalization, they still have the memory of old age. So we can use this immortalized macrophages for our aging-related studies. And has lots of advantages. In particular, we can do genetic manipulation, and we also don't need to every time go back to the mice to get these cells.
Were you able to look at the phenotype of these immortalized macrophages and compare them with the non-immortalized ones, or primary ones, like whether they were more M1 or M2 type, for instance?
Yeah. So, for example, recently we also look at a C-gas activation. It's also very, very similar between primary versus immortalized. So I think it's pretty generalizable. It's not specific for NLRP3 inflammasome. So for this, then we can do this co-culture experiment using this immortalized macrophages. And so essentially we add one piece of white adipose tissue, and then we can activate NLRP3 inflammasome using LPS ATP. And then we add insulin to induce insulin signaling in the metabolic tissue. And then we take this white adipose tissue out to look at insulin signaling. So this will allow us to look at the effect of insulin signaling in the metabolic tissue in the presence of the inflammatory milieu induced by the macrophages. Now, to keep a long story short, when we overexpress SIRT2 in old immortalized macrophages, we were able to reduce the NLRP3 activation. And when we looked at the insulin signaling in white adipose tissues from old mice in the presence of old macrophages compared to young white adipose tissue co-incubated with a young macrophages, there's a reduction in insulin signaling. And when we overexpress SIRT2 in old macrophages, it improved the insulin signaling in the co-cultured old white adipose tissues. So essentially using this co-culture system, we were able to reverse aging-associated inflammation and insulin sensitivity. Now, we also wanted to test this idea in vivo. So we generated an aged mouse model. So this is what we did. We isolated hematopoietic stem cells from aged mice, and we transduced them with either wild type or mutant NLRP3, which essentially it's a mutant NLRP3 mimicking the deacylated form of NLRP3. And then we transplanted them into easily irradiated aged mice to reconstitute their blood system. So essentially, we generate this aging mouse model. So the rest of their body are the same, are both old, except their blood system. They are also from old hematopoietic stem cells, and except expressing wild type or the mutant NLRP3. So we were able to show that. So for the mice, the old mice expressing the mutant NLRP3, they have a reduced IL-1, IL-18 in the plasma, and they are also more glucose sensitive. Okay, so I will just very quickly summarize what I talked about today. So I talked about a mitochondrial metabolic checkpoint that regulates stem cell quiescence and aging. And we showed that this mechanism is conserved across tissues and has therapeutic potential. And I talked about a mouse model that developed spontaneous fatty liver. And we also proposed the model that ER stress suppresses IGF-1 signaling, contributing to fibrosis during the development of fatty liver. I also talked about an in vitro model for studying aging-associated inflammation, and we are extremely excited about it because it's very easy to use, really simplify lots of work we have to do. And finally, is aging reversible? I will show you some evidence that stem cell aging may be reversed, and some aging-associated physiology, such as the inflammation, may be reversible. And I'd just like to thank all the students and the postdocs who did the work very quickly and the funding sources. And thank you so much for your attention and for giving me the opportunity.
Thank you very much, Dr. Chen. It was a really exciting presentation, and I really enjoyed getting, especially the Sirtuin stories. Do you have any other questions, either from students or from anyone online?
Yeah, I just have a quick question. Just asking whether if you have tried bone marrow transplantation or hematopoietic stem cells that are overexpressed in sirtuins, and see whether if you transplant them in mice, whether they affect mitochondrial metabolism or any other phenotype.
So, and looking at it more broadly outside of the hematopoietic tissue, is that what you were saying?
Yeah, you take like a bone marrow stem cells from old or from young, overexpressing the sirtuins, and then transplant them into old mice, for instance, and see check the phenotype or the integrity of mitochondria or something like that.
Yeah, so we are doing something very similar to that. So one experiment we did was actually, so the opposite experiment we have done. So example, we transplanted wild type and 32 knockout hematopoietic stem cells, and then we looked at glucose homeostasis. Yeah. So in that case, we saw that the one with a not 32 knockout hematopoietic stem cells have glucose resistance, insulin resistance, sorry, and glucose intolerance. And so we have a 33 transgenic mice, and so we did the experiment you suggested, and we are currently characterizing the mouse model.
Great. And if the cells are like labeled like fluorescent or something, you may also be able to trace them and see where they go and which tissue they regenerate and all these things. Yeah.
You mean still in the HSC transplanted mice?
Yeah, same thing. Yeah, just
yes.
In addition to overexpressing sirtuins, if they are like labeled like a GFP expression, so you can actually trace them and see where they go.
Yeah, that's a great idea for sure. Yeah, that's a really good idea. We should have done that. And I hope someone supports the experiments. We need these are all very expensive experiments. Are you interested in collaborating?
Yeah, thank you. Very interested. Yeah, thank you.
Yeah.
All right, well, that's all the questions. Then we will go ahead and let everyone get back to their experiments, their day. Thank you again, Dr. Chen. It was a lovely.
Thank you, Emily.
Thank you so much.
Yeah, thank you.
Thank you.